Hey everybody, I'm happy to see you this bright morning. Uh, Let me say a couple of words about me before we begin. My name is Oleg Prilutsky. Currently, I am a head of the Laboratory of Natural Ecosystem Monitoring Biosphere Reserve of National Academy of Agrarian Sciences of Ukraine. In quite recent past, I used to work as associate professor at Karazin University in Kharkiv, and I'm a biologist. Uh, my focus, my topic is fungi and fungal ecology. As a biology student, years ago, at my first term at university, I got C for fine math, and I used to genuinely believe that I've never, ever faced math in my life. So probably in that perspective, I'm extremely different comparing for the most of you presented here. But what's the reason I'm here? I'm a practitioner. I'm using technology to solve biological problems. I'm not a mathematician. I'm not a real data scientist. I'm not a computer scientist. I'm a practitioner who wants to solve their problems. And to do so, I need to reach some technology. And probably the main, our group's the main current topic is producing of machine learning derived global map of fungal ground soil fungal regions. That was a long journey. Now we have some approximation to this goal, which not a final, but right now using molecular tools, DNA metaprocoding from derived from soil, and machine learning, just pretty ordinary random forest, we achieve a high resolution, fine scale global map of mycorrhizal soil fungi for the whole terrestrial, almost whole terrestrial part of our planet. And let me use a couple of next minutes to introduce the idea I want to present you as a possible project for some of you. My very first experience in machine learning was a crop decipher, like were mentioned many times in this the previous day. That was absolutely ridiculous comparing to the level of crowd deciphering we are doing here, and the representatives of agro companies presented you yesterday and the day before yesterday. That was a child game, but that was the first, my personal first experience to get to know what situated where based on very little data. Based on the data we have in one region, trying to project those results to another region. It was almost completely failed. Years passed, and we made some steps towards more accurate prediction of land cover, land use, and vegetation in the land. Our team produced maps of natural habitats for some parts of Ukraine, and not Ukraine only, based on the botanical data, based on field survey of plant species composition. We were quite modest in choosing methods. That's almost exceptionally ensembles of decision trees, like random forest with pretty simple tuning. And under the hood, the moderately large amount of field survey data. But recently we faced the problem. The problem is in Ukraine, we have a lot, unfortunately, of territories unreachable of us. Temporarily occupied territories, heavily mined, territories close to the front line, where we cannot get additional ground truth data, additional data from land surveys, which we can use to train supervised classifiers to get to know which vegetation, which land cover, which habitat types are there. We attempted some experiments, some trials to overcome this problem. We tried to reconstruct the terrestrial, newly arise terrestrial habitat across drained Kakhovka Reservoir. We tried to merge, to fuse pixel and object-based classifications, supervised and unsupervised classifications methods to achieve a habitat map for Scania Nova Biosphere Reserve, which is totally beyond our reach right now, and where we cannot collect more data. And those examples are examples of something which people experienced in supervised classification would call ridiculous, because everybody knows that for supervised classification we need solid ground truth data, which we do not have. Our training data might be ridiculously small. In this map, we can see the territory of the former Kakhovka Reservoir, and the small dots like, I don't know, dung of flies, which are the ground truth data. It's almost invisible on this scale of the map. But we are extremely interested in modeling land use, land cover, and habitat types across such kind of territories. So that's exactly what I would offer some of you who will be willing to participate in this project to accept as a challenge. Habitat mapping based on extremely sparse ground truth data. It's hard, ridiculous, and almost impossible. That's why it is interesting topic as for me. But let me get back to our today's topic. And a second ago, I told you about applying machine learning for very poor ground truth data. And the rest of the time today, I would like to spend to talk about actual ground truth data, how it should be look like, especially about biological diversity. We are talking about data a lot during the past couple of days. Yesterday, Joseph told Nam a lot about, for example, Earth observation data, like Sentinel-2 satellite missions. We're learning about data collected in by agri companies and fields and so on and so on. What's the difference, fundamental difference of biodiversity data from most of the data you are working as a data scientist? Biodiversity data are extremely vague. We cannot even formulate what is biodiversity straight. There are tons of definitions of biodiversity, and because there are many of them, any is okay. And I would pick this one: genes, populations, species, and communities they form. This thin living layer over our stone planet, before our planet and huge, vast, empty space. Why biodiversity matters? This tiny living level provides us a functions which we all are totally relying on our life. It provides so-called ecosystem function. One of my main topic is soil microbiome, and I love this picture about life in soil. Like imagine you are in the soil, not buried, like soil of normal soil organisms. you're looking from the bottom to the upper direction. You can see a whole lot of living things, worms that plants that produce organic materials, that sink chemicals, carbon, nitrogen, oxygen, hydrogen, and produce something new which we all rely on and all life on Earth. We can see a lot of organisms which consume those components and reproduce them further to other organisms, which are transfer it further to other, forming a long chain of communication, providing whole lot of nutrients in energy cycle in this thin living layer, determining in this case climate, food, environment for all living beings, including us. It's impossible to live on Earth without biological diversity. One of my favorite analogies is with the mechanical clock. We all well understand that each part of the mechanism is important. There is no unimportant parts. We can just put only, I don't know, arrows on the clock and say, okay, it shows us time, so it is useful part. All other parts we can get rid of. It doesn't matter which else except arrows in our mechanical clock. We all understand this well. Unfortunately, a lot of people don't understand this applying to biological diversity. They think that there is important organisms, food sources, vectors of hazardly infected diseases, I don't know, pests. And the rest is unimportant and doesn't really matter how it be. This sort of hardly misleading. So, understanding, quantifying, and measuring biological diversity is necessary for produce any other intellectual outcome related to their life on Earth, including producing food, which are the main topic of this school. Any of us has some intuitive understanding of diversity. When we look at this middle, we can say that there are a whole lot of different plant species there. So probably it is diverse community. If we look at the moon, we can infer that there is no such a lot of species in this system, so it's less diverse. This is fundamentally true, but for us, for you as a data scientist, we need not just intuitive understanding, we need quantitative measure of data to work with. So the question I would like to talk today is how we can formally quantify biological diversity to use it as a data for data science applications. And the simplest possible answer how we can describe biological diversity in formal terms would probably be like this. Single unit of biological diversity is the fact that some biological species has been found somewhere at some point of time. So, which species, where, and when? Sounds pretty easy. What's the problem, sir? The main problem is biological diversity. Comparing to, for example, my second favorite part of the data, Earth observation data, is that the biological diversity data originate from extremely wide range of data sources, which implies the extremely wide and hardly standardized range of the way biodiversity data might be formalized. We can get those kinds of data from direct observation. Some went somewhere and observed something. We can get some biodiversity data from historical nature collections, museums, herbaria, and so on, which preserved and conserved for a long time. Recently, for the long time, that was two main sources of biodiversity data, but recently there have been an explosion of additional sources. We get an opportunity to obtain rich biodiversity data from the machines. Photo and video traps, automatic devices that capture animals or any other moving subjects. Drone imagery, photographing of the biological objects from the above. My favorite one, genetic data, metabarcoding. We can get a handle of soil, and after some not so extremely complicated and expensive manipulation, we can get 1,000 of biological species of microorganisms, soil fungi, worms, little animals, bacteria from this handful of soil. A ton of information. Citizen science, social media and different citizen science platforms where usual people post some information about, guys, look at this. I found this nice mushroom. How it called? Can I eat it? All of this arose endless way to obtain the primary data. But what's even worse, on the other side of the coin, there is as many ways to preserve biodiversity data. It might be some handwriting in some field notes or herbaria sheets, labels on the natural collections. There are plenty of living cultures especially for microorganisms. It might be microscopic slides or any other half process information. And now, due to the rise of molecular methods and citizen science data, there are more and more biodiversity data occupied by digital records. In some digital databases, which contains molecular sequences of barcodes of some species, or protein sequences, or any other information derived from molecules. So a whole lot of this form an endless range of the way biodiversity data might be stored. And third part of this problem is that biodiversity data are heavily linked. It don't exist on its own in a vacuum. Some fact of species occurrence actually link with a whole of related pieces of informations. Informations about biological interactions with other organisms. Some organism is a predator of some other organism or a prey of some other organism. Some fungus is a parasite of some plant, and some plant is pollinated by some insect. And those insects have its own parasites, which has its own hyperparasites, and so on and so on. Interactions form the multidimensional networks of biodiversity data. The source we get those data arose a lot of the way to preserve information about the single biodiversity events. Genetic data, occurrence data, behavior data, some facts of behavior of a single living organisms. We have a lot of information about species traits. what functions some organisms can have. Measurements, the distance of flying or dispersal, the birth rate, the speed of reproduction, the probability of local decline of, and so on and so on. And of course, we have information about habitats and communities, which actual biological systems the organism forms. So the problem, comparing to, for example, those observation data, is that to formalize all of this mess, we need to put a whole lot of efforts before we get ready for some kind of analysis. This such an example. There is a plant, Australian, nice Australian plant, Banksia serrata. I saw it just in botanical garden, but it's nice. There is some information about biology and ecology of this plant. There is some organisms directly linked to this plant, like fungus parasite or pest insect. And beyond the real world, there is a whole lot of information you need around this plant. Morphological, physiological features. The species linked with it co-occurring and same communities. Our understanding of the place of this organism in the tree of life and possible misunderstanding from the previous era, which we call synonyms and taxonomical issues, when scientists rethink the position of a species in a tree of life and change their understanding of the linkage between different species. And all of this data might be stored in different formats, in geospatial aspects like a map, in literature like text, in genetic and molecular banks, like a sequence of base pairs or some other molecules. And of course, this problem was attempted to be solved. There are some databases. Each of them is trying to formalize some piece of biodiversity data, and these 6 are not a full list. Of course, it's probably largest or most important. You can see that, for example, Global Biodiversity Information Facility, GBIF, my favorite one, collected more than 3 billion pieces of information about species occurrences. Catalog of Life now hosted more than 2 million taxonomy units, information about a single biological species and its relation, its position on the tree of life, its linkage to the other species. Such a source as a Biodiversity Heritage Library contains more than 62 million pages of textual biodiversity information which have been published in literature. If we get to the molecular side of the view, widely known NCBI on gene bank, as for yesterday, consists of 4.7 billion nucleotide sequences, which can be translated as occurrences of individual organisms. And so on and so on. So you can see that the purpose of this slide is to just show that the amount of data already is huge. And the span, the coverage of those data looks impressive when we look at space view or something like that. For example, Global Biodiversity Information Facility covers with species occurrences almost all over the world at least for terrestrial territories, probably for some oceans. A bit bureaucratic. But is that solved problem? Unfortunately, no. Because, first of all, biodiversity data are extremely rapidly growing. For the last decade, even 1 decade, it multiplied 4 times comparing to the whole previous era of human investigation. As a small digress, just to entertain you, most of the time for the previous day we spent talking about artificial intelligence and some other techniques. So I just want to show you some little but meaningful case of using large language models just to mine biodiversity data. A whole lot of biodiversity data have been published in scientific literature, so encoded in human text, in words. This data about species occurrences. I went somewhere and found something, some about interactions, and I observed, wrote some famous scientists from the past that some insect is, I don't know, feeding on some plants. And also I measured the length of legs for this insect, for example. So there are some numbers. All those encoded in human words, and it was a real pain in neck to decode it using all the complexity of human languages and large language models. For several years made an exceptional progress in that case to parse the human written text into the so-called taxonomic events or taxonomic treatments, like information about something has been found somewhere at some time and connected with something else, and something have been measured. Such resource as Platzi, hosted in Switzerland, hosted the data source called treatment bank for such kind of taxonomic treatments, as they call them. And you can see that for yesterday, it processed more than 85,000 of scientific articles and extracted more than 11 or 1 million of individual treatments. That's just to entertain you a bit before we get back to the main topic, how difficult to deal with biodiversity. And another issue with biodiversity data is that the data is not enough to deal with biodiversity data. Because for the reason biodiversity data is highly diverse and complicated, we need to another part of information to translate us or machines, which this kind of data means, so-called metadata. Data about data, translator, interpreter, who describe in which manner the biodiversity data are codified and stored. And creating those kind of data is a challenge itself. I guess most of you are familiar with the concept of FAIR data. It's an abbreviation of findable, accessible, interoperable, and reusable. For nicely sounded words. What in application to biodiversity data it can mean? Findable, that the data have to be findable, have to be allowed to be explored both for human and for machines. The most direct way to achieve this if we are talking about biodiversity data, is to pursue persistent identifiers and provide sufficient metadata. Accessibility means that the data must be understandable and transparent both to humans and to machines. In order to achieve accessibility for biodiversity data, we have to use some trusted and transparent, well-documented repository for data. Unfortunately, traditional ways to store biodiversity data cannot be treated as accessible. Here in Lviv, we can a huge biological collection, natural history museum, but are they accessible for any of you? I hardly say so. For most of you, those materials are not accessible, not to mention to your analytical purposes for some machines or agents or something like that. It's stored in a way which cannot be communicated from outside. Interoperability means that we provide high standards of both data and metadata, which can be codified and turned for both ways, from human to machines, from machines to human. And reusability means more legal side of the coin, using of clear licenses clearly postulated the way data can be treated. And the first and foremost step here is the codifying the different aspects of biodiversity information. But for this end, several biodiversity data standards have been developed. That's pretty boring stuff, but extremely important one. The most well-known and the most widely used, especially in ecology and biodiversity informatics, is so-called Darwin Core standards, which are developed by which is developed by the non-commercial, non-governmental organization abbreviated to called TEDVIC. This is you can visit this web page and get familiar with the logic of Darwin Core. It's just a controlled vocabulary for almost any possible piece of information related to biodiversity, especially when we're talking about species occurrences. How can we, in controlled vocabulary, in some limited variants, describe the, for example, time of occurrence? Which standards should we use? Have to we use? Yeah, I would say. How informal way we can describe interactions between species, host, pest, predator, prey, and so on, pollinator, and the plant which are pollinated, and so on, and so on. It's human readable and designed to be manually processed, first of all. So actual human being, actual biologists who produce or digitize existing biodiversity data might be able to read it, to understand it, and use this controlled vocabulary to prepare biodiversity data at the first stage before it gets fully teachable. On the other hand, machines have to understand those vocabulary in an unequivocally way. Probably it sounds like a bit of far from reality. Let's look at some example. Classical botanical data, vegetational survey. Some natural communities, some human botanists got there, make a plot and fix all the plant species which they observed. The primary way to store those biodiversity data is to paper blank sheet where linear names of plant species are stored with the information about their projective coverage. Which percent of this fixed area, fixed size plot are covered by a particular corresponding plant species. That the way plant surveys have been done for centuries. How we can translate it into a controlled digital object? Darwin core standards allow us and offers us its own data, its own way to digitize different piece of information with this data, which calls the Darwin Core Archive. Technically, that's the zip archive, and it consists of some core information, which are all tabular data, or tabular. I don't know how to pronounce it correctly. It's like spreadsheets. It's nothing extremely complicated. It's human readable spreadsheets, tabular data, but linked in a specific way. In this example, in the core of data is so-called event core, which describe in controlled vocabulary the plot, where, when, who, in which condition, all the information about this plot, geographical coordinates, date, and so on, methods applied to survey. With the core, link some other units, tabular units, some other spreadsheets. For example, occurrence extension, which provide row by row data about individual species. Which species, each has unique identifier inside the archive, linked with the unique identifier of the which event. And some basic information, or not basic, or extended information about this species. Measurements exactly, like age, sex, I don't know, what else we can say about species at all. A whole lot of things. But what if botanist measured something? I don't know, leaf area, for example, or number of blossoms or flowers. Okay, there is another tabular unit in another spreadsheet, measurement or facts, which has its own unique identifiers linked to unique identifiers from occurrences, and store the data about leaf area, of number of petals and blossoms, and so on, whatever they measured during the survey. And that's not the full list of possible extensions, but those left part of the scheme is just data. We have also metadata. Metadata, Darwin Corp's archive allows several ways to store metadata. Most important are human readable RTF, and machine readable XML, which are interoperable and can be easily translated into one another. And all of this packed in a single zip archive and presents a unit of biodiversity information which can be achieved, which can be downloaded by human and can be read with eyes as a tabular data, which can be achieved by machines, and can be processed as a structured unit of information. In other words, it's like one-to-many SQL database, where each object has its own identifier, and there are some range of tables. Question.
Oleg, can you please clarify? Can you please clarify if there is some occurrences about Ukrainian species in this archive? And please clarify, like, what location, what specific species are there?
Thank you. Yes, of course. There is, last time I checked, there have been 2.4 millions of informational units, species occurrence, just for the territory of Ukraine. And that's not the structure of a whole database. That's just the way data are stored. There are plenty of archives. I don't have a dedicated slide in this presentation. After the lecture, I can, if you are interested, show you some Ukrainian part of this data that are huge, and there are several dozens of data publishers, institutions. There are hundreds of data sets individual, which dealing with different taxonomy group, different ways of collecting data, and so on and so on, and thousand upon thousand, I don't know, I can't collect individual Darwin core archives. But the unified structure of data in metadata allows us to, if we're talking about GPF or something like that, allows us to make a query to the global database and extract any piece of this information in our own Darwin Core archive. I can search for particular species which have been founded just in Lviv region, just between 2021 and 2023. I don't know, just by some, I don't know, Petrenko, what it is. So I can formulate the query as for any database and can get structured my own Darwin Core archive for this data. So please.
Does the structure, this Darwin Core archive, include photos? And is there any moderation for those archives of the internet? So, because I feel there is a room here for some incompetence from people who upload this information. So at least I can check photos, check information, and moderate this information so we can use it.
Thanks. I was going to pay some attention for this question a bit further, but in short, Darwin Core Archive and any other, this is just one of the biodiversity standards, not the single one. All of them aimed to embrace all the possible way to store and translate biodiversity data, including photos, videos, movies, living beings, living organisms, and so on, through the link. So as this spreadsheet linked to each other, we can link image, link video, link anything which we can imagine. And to the point of quality control, that's another point, but let me say about it in several slides later. On top of the control data structure, the way to achieve control data usage is to produce stable identifiers, and it's extremely popular hashtag in biodiversity informatics, CiteDOI, assigning digital object identifiers to any piece of information, including data sets, including individual query, individual species, and so on and so on. So, for example, a data set can have its own unique identifiers, which can be used as reference in scientific publishing, and processing those references can control how many times which biodiversity, which unit of biodiversity information has been cited. So, two issues. It wouldn't be so easy and so relaxed as it looked like. Of course, there are a lot of them. First and foremost, as for me, is taxonomy, because that we are human beings, Homo sapiens, from the class Mammalia. It's okay with us. Not so with the most of the living organisms in the world. Most of the species in the world are still undescribed, first of all. Among described species, there is constant flow of understanding where which species must be must situated in the tree of life. So taxonomy, species naming, and understanding of species relationship are constantly changing, and that's a disaster. There is a kind of biologist, systematic biologist of taxonomies, to spend whole its life to understand the position of tree of life, some very, very little probably groups of living organisms, and for some of them there is no single dedicated specialist in the world, especially for many kinds of insects. So taxonomy is constantly changing. What can we do with that? It means the name we use today might be invalid, wrong tomorrow. There is digital database just to control usage of names. For example, I am fungal ecologist. For fungi, there is a famous resource, index fungorum or species fungorum, which, by the way, sustained by few images, and I don't know how what pharmacology would look like if tomorrow it will be shut down. Different taxonomical sources might be mobilized in a single, more or less coherent structure. For example, GBIF tried to sustain its own so-called backbone taxonomy, which is a consensus of many other digital taxonomies and which is changing all the time, exactly once per 2 months. So in the 2 months, quite probably the green toad would be named differently. And you can see in the left that all the name below Pilophilus erdibundus, the Latin name of green toad, is a name which were valid at some point of time, or were not valid but meant the same. And biodiversity informatics must understand that something which were called Ufa ridibundus by Pallas in 1771 is the same which current modern scientists call Pilophilus ridibundus in some other part of the world. It's extremely complicated task, and there is digital backbones, so-called, who which try to make a compromise between existing digital taxonomies. And species name mention is not unfortunately controlled way, and I hardly imagine the world if it's achievable at all. The world when we have full agreement of how we call species and how we put species on the tree of life. Even more troubles arise with the rise of molecular data. There you can see, let me step back, for example, plants, fungi, animals. There are some number of described species, but as we know, the around half of plant species are just undescribed. We know about them from the unique genetic markers, and we don't have names for them. And for fungi or for animals, especially due to insects, it's even worse. We don't know most of the biological species. And since the most of biodiversity data now comes from molecular research, the fraction of this unknown dark diversity, so-called, we know dark matters in the space, which we can't see, but it exists. So there is dark diversity, which we can't name, but it actually exists. And rise of genetic data makes it just worse, because the way we name species, which Carol Linné established 2,500 years ago, cannot today answer the question which species occurred here, where? And we continuously go from normal Latin nomenclature we taught at school to the so-called operational taxonomic units. Unique, specific sequence of genetic information indicating a single functionally and reproductionally single species which we can't even name. And in some resources, it happened to even incorporating such genetic information to the actual taxonomy. For example, for fungi, there is a resource called United database, which stored, I don't know how many, millions of so-called species hypotheses, which are substitution of linear species in a world where molecular data are growing fast. And all of these species hypotheses have its own digital object identifiers to ensure interoperability, and its own formal name, like SH0789009.10FU. It means nothing. It's not a accustomed way to name species. But what else can we do if we have species which are still undescribed, but we know for sure it exists just from genetic information, just from the handful of soil we dig somewhere, or I don't know, glass of water in some small lake in the vicinities of Lviv. Assigning digital object identifier to each hypothesis of taxon is probably the most most solid recent way to process those data. And since the lot of biodiversity data are stored, processed, acquired in a different way, there is a huge problem of data duplication. Some plant species have been collected somewhere by someone and stored in herbaria. Herbaria have been digitized, and we have a digital piece of information, digitized specimen. We have a photo, which is another piece of information, probably made by someone else in some citizen science resources. We can have extracted DNA, citation in literature, scientific, and so on, many kind of information which can be individually and independently digitized. And such kind of data duplication artificially increase the amount of data without adding useful information. So finding links between different pieces of information of a single species occurrences, a single taxonomic treatment, single real event in the life is a huge informatician problem. What you asked about quality control. One of the largest source of biodiversity data nowadays is citizen science data, where anybody can use their smartphone to take a picture of something and post into the internet. There are resources, dedicated resources for citizen scientists, which implement its own quality control techniques, like requirements for proofs, like photo, video recordings, sound recording, and I know for birds, for example. Some require voucher specimens to be stored in some collection and checked. I can't, I have not so much time to describe all the way quality controls, but in short, it's not so good with quality control and biodiversity information realm. Because the reasons we described it about a huge race of data and low problems with data unification. A huge problem poses the fact that our digitized part of biodiversity data is not the mirror of the real world in terms of representation of different taxa. For example, in this picture, we say the bug, which is huge, and an elephant, which is quite small under the much bigger family. The size of an organism represents the number of described species of different parts of biodiversity. And you can see that mammals are not in the leading role. But on the other hand, in the digitized part of biodiversity data, vertebrates like mammals, birds, reptiles, amphibians, and so on, fish, occupy much larger part than the insects and the nematodes, fungi, bacteria, viruses, and so on. So we underestimate a huge part of the biodiversity, and we know about that. And we still can do something with that. And to wrap it up, a couple of words about how biodiversity informaticians envision the future of digital biodiversity data. This is one of the many weather in interactive weather forecast map. We talked about weather forecast and weather modeling 2 days ago, and I hopefully understand the origin of this map. There is some data collected on the ground in some meteorological stations. Stations are not covered across all the world. And then, based on those data, there is some spatial temporal models which predict how some environmental variables, temperature, wind speed, humidity, rainfalls, and so on, would behave at some point, at some moment of time. Yes, we all accustomed with map of weather forecasts. What we want to achieve, the digital imprinting of life, in a way, in a forms of maps, in a forms of some dynamics representations. It might represent different forms, but anyway, to something that anybody can just open, look at, or download if you are data scientist are willing to process it in machine way, not just with human eyes, and get some data for any point, for any time moment, for any point in space and time, and make some inference, make some decision if you're a decision maker, or calculate some metrics, or understand something new and important in your perspective. Our team last month presented Ground Underground Atlas, which is a quite simple representation of the diversity and species richness of soil fungi detected by metabarcoding of soil globally and modeled globally using machine learning techniques. It now applied for a quite small part of fungal work for ectomycorrhizal fungi, those who are extremely important to sustain tree and shrubs plant, a perennial long-living plant, which we call tree and shrubs, and arbuscular mycorrhizal fungi, which are obligate symbionts and direct, how to put it, organisms which grass and annual plants are totally indirectly related to consume some chemicals, including nitrogen and phosphorus. And this map is static. So no dynamics comparing to weather forecast. You can just look at it and make some inference if you want. But the idea some have at some point of time achieved the state when almost all biodiversity data are findable, accessible, reusable, and interoperable and reusable, encoded in both human and machine readable formats in controlled way, allowing to easily access the data and use the data in models. What models it can be? As a couple of examples, I have a couple of minutes left and a couple of examples have people envisioning, and both of these examples are actually have its working prototypes. It's not just my hallucinations. Decision maker in forestry all around the world face the problem. They somehow need to do logging. Need to cut trees to get wooden materials. That's how forestry works. On the other hand, we know that just cut all the forest leads to tremendous degradation of ecosystem, erosion of soil, and new forest won't grow on this place. It would be very bad, not just to biodiversity, humanity, local communities. It would be just for business. So we need somehow foresters, needs forest management managers need somehow to know how to cut which trees they can cut to minimal loss for ecosystem functioning to sustain sustainable forest in this place. How they know that? They hired scientists, scientists go somewhere, made some research, write some reports, they analyze it. It get a whole lot of time, and due to inconsistency in methods, in groups working, in target parts of biodiversity and forest ecosystem, it turns to be quite unreplicable. First of all, and secondly, it turns to be it takes quite a long time. So the idea of this so-called forest digital twin, like twin of the real world, but in digital realm, is to provide decision makers in forestry the working and constantly updating model which consume data about forest structure derived from field surveys, from airborne surveys using UAV, using LiDAR footage, and so on and so on. Data about key groups of forest biodiversity, which forests heavily rely on to produce a sustainable life cycle of trees. Assessing of forest parameters as biomass, as productivity, and so on, assessing the most vulnerable parts of forest communities, which are sensitive to logging, to forest management practice. That's fitting the biodiversity model, which predict for each pixel, for each part of the space, the possible outcome for biodiversity of forest ecosystem due to some scenario of forest management. And so decision makers have like a computer game when it can press the pixel, move the slider, change the amount of timber which will be collected, and in almost real time, getting information about which impact it can have on forest ecosystem in a year, 2, 10, 20, and so on and so on. And then make a decision about fitting the practice, change the decision, try another part of the map, and so on and so on. Another work in digital twins relies with genetic data and undersampling. We despite the fact we have a lot of biodiversity data derived from environmental DNA, soil, from food, from plants, from water, and so on and so on, we still have a huge part of the world unsampled, where no one even went, even gone, no one have ever collected some such kind of data. And we well known, well aware that making predictions like this is heavily impacted on the distribution of primary data. And there is no point in so primary data in this map, but I can assure you they are not distributed across the planet. In the Sahara desert or I don't know, some in ocean islands, there are probably close to zero data points comparing to the rich, well-developed countries of Western Europe or US. So how we can quicker improve the model? To improve the model quicker, we can understand undersampling. To understand undersampling, we can just after we feed the model. So when I am a, say, field biologist, and I'm going for some expedition, some field survey, I need to understand where I have to go to achieve maximum results. To understand that, I might consult with my colleagues, I might stay, think, I don't know, read scientific literature. It would be highly subjective and would take pretty long time. On the other hand, I can achieve standardized data of genetic biodiversity from GB, for example, calculate based on those data community metrics, which we are going to talk tomorrow morning. Based on metrics of biodiversity, I can recalculate the map of priority survey to understand where I have to go to get new piece of data to improve my model the most. And this way, in a quite short time period and more important, reproducible way, I can tune my individual research to achieve more results in less time. Both of those prototypes are working, but the idea behind digital twins and biodiversity are growing, and I hope we will witness the day when biodiversity information will be so easily achievable and understandable as a map of weather forecast for today, for example. And to this end, the common efforts of biodiversity researchers and informaticians might have to meet. So that's all from me for today. I hope I at least frighten you up about how complicated the topic of biodiversity information. And I hope I convinced you a bit that biodiversity is extremely interesting part for applying of information technologies, which are strongly expecting and are waiting for you to put your hands in it. Thank you so much. Open mic, please.
I have one more question about the digitalization of data, especially Ukraine, for Ukraine. How much data is digitalized? For example, we talk about data needed for your research. Any activities of digitalizing, and I mean, do we need to digitalize everything and like that?
As for now, talking about Ukraine, I don't know, it's hard to make a direct estimate of which part of biodiversity data are existing, collected, and stored somewhere. Biodiversity data have been already digitized because they are not digitized. And we don't exactly know how many biodiversity data we have. We know that there is a huge and profound research institutions, like in National Academy of Science, there is an Institute of Botany. And I know that there is a National Herbarium of National Academy of Science in the Institute of Botany in Kyiv. But nobody knows, including the researchers from the Institute of Botany, how many specimens of plants are stored in this herbarium, because they are not digitized. They tried to make some paper cataloging in some journals, in some blank sheets. With time, it have been lost, most of it. And for now, we don't even know which amount of data we have. It's just quite wild, there are rough estimates. As to the already digitized part of this work, we can more or less correctly this estimate because it's digitized and preserved in interoperable format. As I said, for example, for GBIF, just for GBIF, not for all possible way to store biodiversity data, we have close to something more than 2.4 million of data points, taxonomical treatments, taxon name, place, time, and some other information possible. For right now, metabarcoding efforts on the rise. There are two projects of genetic identifying of biodiversity underway, which I know about, and probably there is some I don't know about. For example, our team collected 2 years ago 225 soil samples across Ukraine, and now we have the result of metabarcoding and know about 15,000 of operational taxonomic units of soil fungi. We work just with fungi. There is an attempt to do the same for water, for Black Sea Basin, Northern Black Sea region, for freshwater ecosystem in Dnieper, Siversky Donets, Southern Bukh rivers, within barcoding, so-called European project Barcoding of Life. But I don't know about results of this project. So we can pretty we can precisely say how many records. If I go to gbif.org, I can say you after the lecture exact number of data points for Ukraine. But I expect, based on my experience and communication with other scientists, that that's just a small fraction of what can be digitized. And moreover, digitizing of existing biodiversity data is one win. And another win is the collecting newcomer data. And both citizen science, molecular research promises to, I don't know, double existing data each year in some log scale. So even though we have much more data than we can process by traditional methods of analysis. In 10 years, the amount of data I expect to be unbelievable larger than we can process traditional method. And my guess that machine learning and artificial intelligence techniques might be extremely in use in quite near future because the amount of data are rapidly growing. And what we do now, we are hardly try to, I don't know, don't get so bad of growing the data. We try to digitize existing nature collections or standardize the way current data collected, but quite not that many years will pass when we face that machines will collected the amount of data any of Ukrainian natural collections were left far behind.
Okay, you mean that we don't need to digitalize existing papers? We can like gather new already digital information from now, like within several years.
I guess we need both because Ukraine, Ukraine in interesting situation. It's so-called it's tragic situation due to the war. But for many kind of activities and for biodiversity research as well, it caused some issues that, for example, we need to understand how the war impacts the wildlife and life on the territory of Ukraine. How can we do that? To do that, we logically need information about how life looked like before, how it looks like now, and how it will look like tomorrow. So we need all of them. We need current monitoring, or we need digitized previous data before they die, because data, unfortunately, in scientific collection, in any way, the data are dying. That's a big problem. Not just after shelling or rocket attacks, data dying just because human stupidity, data are dying with new way to communicate with data arise, and legacy ways have been abandoned. So we need all of this, digitizing previous, monitoring current, and get prepared to process future, what I say.
Thank you for this lecture. Although you still need to document not existent species, how do you know by sampling soil if it exists or not? Because the leftovers of any species can be in the soil, but it is not a fact that it still exists. And at least one data point will be not 100% sure that this existed or not.
All positive problems. Excellent question, thank you. We still heavily rely on DNA, which are quite stable molecule, and you're quite right. The DNA molecule in some situation can be preserved for years after organism's death. And that indeed is problem. There is a solution for this problem, which are not so widely used because it's methodologically complicated. But I guess in several years it will be solved and easily achievable. It's used RNA, ribonucleotides, which are the replica of DNA when the organism produces proteins. So RNA exists only for organisms which are alive, or will be alive 5 minutes ago. For now, we just technically, the problem with RNA that it is quite unstable. So collecting, preserving, processing of RNA are still just technically laboratory complicated tasks and more expensive, much more expensive. That's another limitation of this. So yes, we are aware of this problem and try to solve it. For us, for microbiome researchers, it's a bit easy. We can relax a bit this assumption because soil microorganisms, both fungi and bacteria, are live a strange life, and the exact border between living life and death is not so obvious as for us, for example. And fungus or bacteria might be inactive for years or decades and not dead. So yes, we know about this limitation. There are a whole lot of issues with molecular research. This is one of them. There is a solution, but unfortunately, this solution has its own limitations and methodological limitations. But great point. Thank you. Maybe? Thank you for your attention.
Online for questions.
So, okay.
You can unmute yourself.
I think it was long ago. I think your risk is kind of like afterward.
What can I do?
You can unmute yourself, I think. Can you? I don't know. So then ask the question.
All right, I have a question. How we do discover data that is unknown?
What do you mean, Evgeny? Unknown, we can't discover the unknown.
I mean,
Because-
you just mentioned the data in the geographic like institutions that is not digitized, and you can't. Did you say that we can't estimate how much is there like stored in those paper archives?
Maybe how I assess it, that so much unknown data if you do not know about them. Maybe.
Probably. Thank you. We have some indirect assessments, indirect estimations. It strongly depends on which kind of data we are talking about. If we're talking about scientific collections, for example, how we can know which part of scientific collection have already been digitized? That's relatively simple task because any scientific collection has its own belief how many specimens they are stored. Is this belief a truth of true or false? That's another question, but we can just start from this point. And when some natural history museums say they have 1 million specimens, okay, we can start with this point and aim to digitize approximately 1 billion of species. That's like human behaves. If we're talking about data in nature, like how do we know the fraction of undescribed species of insects, say? Yes, if they are undescribed, how can we know that they exist? I was going to talk about this tomorrow morning more, but there is pretty simple techniques to predict the unknown part based on already known. For example, assessing the frequency of rare events. For example, we collected new information about species, and some species which are more frequent, more abundant, happen to occur again and again and again and again. And some species which are less abundant happen to occur quite rarely, once, twice, in a big subset of data. And analyzing this dynamics of data accumulation, we can numerically estimate the unknown part of the data. And by the way, that was a great gift of Alan Turing, famous mathematician who applied this method first to estimate Enigma Nazi cipher machine, to estimate unknown part of the message of Enigma machine. And now it applied to biodiversity data, I believe not just in biodiversity data, it's a general method. So it depends if I get your question right, it strongly depends on which part of biodiversity we are talking about.
Thank you. Pretty much. I would just very interested in forestry preservation and like monitoring wildfires is kind of they use also like the simulations for like forestry. So how do we know how much fuel there is in? Yeah.
I am unfortunately not well aware of how forestry simulation works, but probably you're right. I guess there are a whole lot of methods to estimate unknown part of the data from already known. And I guess there is another quite great room for information theory and practice and modeling in biodiversity science. Thank you again. I guess we need time to rest right now before another activities. Не зрозумів, куди натискати. У мене був диплом бакалаврський в мегадексида. Ну, я давно забув цю історію, звісно, це було таке помилка юності.